of the exam is the same, right? And I did post a detailed announcement from last week that tells you what is in it, on it, and what is definitely going to be on it, and what's definitely not going to be on it. So as far as the formatting, the logistics, actually, let me just go with the logistics just to be on the safe side. The exam is on a Sunday. It's not Saturday as usual. It is on a Sunday, and the reason is simple: is Saturday is devoted, for finals week at least, is devoted for common exams. So some of you probably, if you're taking chemistry or some English, I guess, or some courses where you're all taking the same exam, that's where Saturday comes in. So for those of us who give exams on Saturday, we have to give it on a Sunday for the last one. There are still 50 questions. The timing is also different. It's not at 11 as usual, it's at 12:30. Please mark your calendars. It's Sunday from 12:30 to 2. It's the same room separation, so if you're in A to K, I think you're going to be in 1051. If you're L to Z, you're going to be in 1061. Same format, 50 questions, 40 that are regular, and then 10 that are true or false. And the material is 2 modules, so they're going to be roughly equally represented. So it's not like there's going to be 40 questions from Module 8 and 10 from Module 9. It's going to be about 25 each, give or take a few questions. And it's going to be a mix of questions and problems, because both chapters, both modules lend themselves to that information, okay? So in Module 4, we talked about epigenetics, right? That is a big part. So in epigenetics, what are examples of epigenetics that were discussed in Module 8, right? There was the idea of genomic imprinting. There was also maternal inheritance. There's also trinucleotide repeat diseases. Well, genomic, actually, let me rephrase that. Excuse me. Right, maternal inheritance doesn't really fall under epigenetics. In epigenetics, we really discuss genomic imprinting. We discuss X chromosome inactivation. And these are the big parts, right? So with genomic imprinting, you are expected to know, what is in the notes. Now let me just stress this point too. For this exam 4, you're only going to be responsible for what is in the lecture. Even though I mentioned maybe reading assignments from the textbook, you're not responsible for that. So whether it's in Module 8 or whether it's in Module 9. Okay, here it comes. Yeah, right.
You're gonna find okay.
Thank you. Thank you.
She's gonna get another one too.
Okay, so those not cheap either, right? Right. Oh, okay. Thank you. Yeah.
I'll be in the back.
Okay, cool. Thank you. Thank you. Right. So again, let me just, now that we have the rates. Oh, okay. I was going to use one of these shirts. Thank you. Hopefully it doesn't fall. Thank you. Oh, is that the mic? Okay, thank you. Okay, so we'll start with, since I have, with epigenetics, there's really one major phenomenon which we discussed in detail, which is genomic imprinting. Then we also talked about X chromosome inactivation. Now there's a part about development of specific cell types, and this morning when I was preparing for this meet, I realized that I did not include it in Module 8. These slides were not included, my bad. So what I'll do is I'll put it in an announcement so you don't have to worry about that part. So in fact, might as well put it now and in an announcement. So the third one, which is not in your notes as of yet, is development of specific cell types. Specific cell types. So I'll talk a little bit about each. And as I indicated in other M.E.E.T sessions, I think certainly for the conceptual part, I think what would be helpful for you when you're studying is to basically try to do what I'm doing, which is concept maps. So put down the big heading and put an arrow with what are the subheadings, what are the further subheadings and things of that nature. We'll start with genomic imprinting, right? With genomic imprinting, and one other thing that I need to mention before we go forward is if there are figures that I referenced in the notes, also you're not going to be responsible for the details of the figure. So for example, there's one that talks about how X chromosomes gets inactivated, the role of CTCF factors, etc. You're not going to be responsible for that part. Okay? So just to to stress this. What is also important is to know what are the role of DMR and ICR. What does DMR stand for? So when something is imprinted, let's just go over conceptual. When something is imprinted, right, it means it's silenced. What makes it silenced, right? What changes to make it, may render it silenced or render it not active? What are the major things that happen to it? What groups are attached to make the gene inactive? Sorry? Methyl groups, right? Who said methyl group? That's basically what DMR stands for, differentially methylated region. ICR stands for imprinting control region. So differentially methylated region, imprinting control region. So basically that is one important point to remember for the exam. Usually when a gene is silenced, it is imprinted, there's usually a methyl group attached to it. So that is why these are epigenetics. These are above the level of the gene. These are not mutations because they could be reversible. Okay? So, okay. So that, now what is the enzyme? You also have to remember the enzyme that adds the methyl groups. And then the enzyme is DNMT1. Okay? DNA methyl transferase 1. That's the enzyme that adds the methyl groups. So if you're creating, a concept map or a flow chart, put genomic imprinting, right? There's methylation. Methylation, this is the enzyme that is involved, and it typically acts at the level of the DMR and then the ICR. So there's an imprinting control region and there's a differentially methylated region. Both of these are typically methylated and they are controlling the expression of the particular gene. Okay? So another small aspect that we talked about was we talked about two particular genes really. We talked about H19. Again, this is all under genomic imprinting. We talked about H19 and IGF2. This is insulin growth-like factor 2, and this is simply called H19. The difference between the two is this. H19, I'll get to this point. Now, these are the two genes that we discussed at length when it came to genomic imprinting. And if you go back to your notes, you'll know this. H19 is active in mom. IGF2 is active in dad. Right? So when something is imprinted, what does it mean, right? If something is imprinted, it means it's silenced, right? So if I told you gene X, right, let's not confuse X with the Y chromosome. So let me use gene Z. If I told you gene Z in humans is imprinted in mother, in female, right? That means it is active in males, right? So imprinting means silence. So here, this gene is active in females. So therefore, it's imprinted in males. This gene is active in males. That means it's imprinted in females. So that means mom's copy is inactive for this one. Another difference is that H19 gives you a non-coding RNA, and this one gives you an actual polypeptide. So these are the two differences, and these are really, 1 or 2 points on the exam right there. Please know that H19 is active in mom, so silenced in that, and it gives you not a protein, but a non-coding RNA. This one is IGF2. It is active in dad, not active in mom, and therefore it will give you a polypeptide. Not therefore, it also gives you a polypeptide. Okay? So questions about, there's a part in my notes and in the book that has to do with CTCF. You're not responsible for that. You're not responsible, so any questions about genomic imprinting as an overarching theme? There's also a section on pluripotency factors. You're not responsible for that either. So I'm trying to go over conceptual stuff, and then I'm going to go over some of the problems. But are there any questions about conceptual stuff? Okay. So you remember, you have a few true-false questions. So if a question reads something like this, all genes are imprinted, true or false? False, right? Only some genes are imprinted. Okay, and true or false? A gene that is imprinted in both males and females. False, right? Because that's the whole idea about imprinted. It means it's only copied, only active in one sex and not the other, right? Okay, so let me carry genomic imprinting to its logical conclusion by doing a couple of problems. And we'll focus on IGF2. So let's start with this cross. So if you're told that you have IGF2 times IGF2M, IGF2M. And then here's the key. And I'll ask you the question in a second. And then, actually, let me leave it at that, then ask you the question and see what answer you come up with, or the class comes up with, or someone comes up with. Here's the cross, IGF2 times IGF2 times IGF2M, IGF2M. Remember, M is mutant. Now, remember that the idea of these MEET sessions is I'm going to ask you questions as if you are ready to take the exam, right? So whether you know it now, hopefully you do know it now, but if you don't know it now, this is the type of questions to expect come Sunday. So here's the question. Given this cross, what % of the offspring is going to be happening, right? If you do this cross, what letter would correspond to their offspring?
Don't we not know unless we're given which one's male?
Okay, good for you. So for those two, I'm going to be handing out shirts, but I'm not going to hand them out now. I'm going to hand them out at the end. So can you please also, same to you, please. So if you guys can come back at the end. Right, that's exactly, and then these are not from my budget. These are from the Marshall Center. So we are expected to give them out. But anyway, that's exactly right. We can't answer it because we don't know who the father is, right? So if I were to ask you a question like this, I would have to have a choice E, for example. I would remove one of them, cannot be given, cannot be answered from the information given. We need more information. So then I go back and tell you, okay, you're happy now. I'm going to tell you that this is dad and this is mom. Okay. Now you know that, and by the way, these are the symbols, right? This is the symbol for males and this is the symbol for females. So if given this, what is the answer now? Or you still can't tell? Why are they all normal?
Because the mutant gene is only being imprinted in the mother.
Right, exactly. For this type of cross, with all due respect to mom is irrelevant, right? Because mom will give you an inactive copy anyway, right? Will give you an inactive copy, even if it's normal. In this case, it's a mutant, but it's irrelevant. Mom is not contributing except really the egg here. The phenotype all comes from dad. Why? Because the IGF2 is only active in dad. So in this case, the answer is 100% normal. Right? So, here's another question. Okay? True or false? In the case of genomic imprinting, reciprocal crosses yield the same results. True or false? In the case of why false?
Because it depends on which one it's imprinted in.
Right, exactly. So for example, let's do this one. Let's do a reciprocal cross. What would you expect if it's a reciprocal cross? All of them to be dwarf. Why? Because now dad is the mutant. Mom is giving you a normal copy that is silenced. Dad is giving you an active copy that is mutant. So therefore, they're not going to get the same result. Now, we also talked about, in the context of genomic imprinting, you are responsible for Prader-Willi and Angelman syndrome. Now, these are two syndromes that we discussed. And then here, let me just summarize it so that you get a sense of what to expect. We talked about Prader-Willi syndrome and Angelman syndrome. Here, I'm not going to go to the level of which genes are involved. Okay, I'm not going to ask you, is this a brain RNA gene, or is this a splicing? But I do expect you to know where the gene is imprinted, right? So, and then remember, imprinting means silenced. So the reason we get Prader-Willi, or an individual inherits Prader-Willi, is because of a quote-unquote a problem in who? Right, in that, exactly. Okay? So Prader-Willi, and again, I remember it decades later, think of it this way. Prader-Willi is a result of a paternal deficiency, okay? A paternal deficiency. So what does that mean? And it's easy to remember them, PP, right? Prader-Willi, paternal deficiency. So deficiency means dad did not contribute what he was supposed to do, which means you get a deletion from who? Dad, or you get what? Sorry? Not an addition, what is that called? Not an insertion. So you either get a deletion from dad, which means that, mom gives you a normal chromosome, dad gives you a chromosome that is deleted, or you get, you're almost there. Okay. Or you get two chromosomes from mom. This is called maternal dystrophy, right? So Prader-Willi is a result of a paternal deficiency. Or simply remember it as a paternal deletion. Right? Angelman is due to a maternal deficiency. So how can you get a paternal deficiency? Either you get dad deletion, let me just summarize it, or you get two chromosomes from mom. Right? Here's the exact opposite. Here, you're going to get what? You're going to get a mom deletion or two chromosomes, two dad chromosomes. Now, knowing this, right, while you don't need to know the exact reason for the gene, or at least the exact function of the gene, true or false, the gene for Prader-Willi is imprinted in mom. The gene for Prader-Willi is imprinted in mom. True. Because remember, imprinted means silence. So how can you get Prader-Willi? Because dad did not contribute the right gene. So that means the gene was active in dad. It's silenced in mom. It's imprinted in mom. So getting two chromosomes from mom, you're not getting any active copy. Getting one chromosome and one deletion from dad, you're not going to get any active copy. Questions about this idea? Okay, so that's a Prader-Willi. X chromosome inactivation, there's a lot of detail, but you will not be tested on it. For X chromosome inactivation, you have to know basically what are bar bodies, right? How do we determine the number of bar bodies? and that's pretty much it. No, you have to know the roles of the two genes, right? We talked about only two genes. One was exist and one was 6. That's it. So let me summarize them real quick. Again, please stop me if I'm making sense or if you have questions. So for X chromosome inactivation, you basically have to know the idea of bar body, right? And this is an example under epigenetics. So a subcategory is X chromosome inactivation, the number of bar bodies, how do you determine that, right? And really there's only two genes that we talked about, exist and 6. So exist increases X chromosome, or causes, exist causes X chromosome inactivation, 6 blocks X chromosome inactivation. That's it. You're not going to be responsible for any of the details, CTCS, etc. Pluripotency factors, all that. You're not responsible for it. And then the final category, which again, it was my bad, I did not include it in the slides. But you are going to be responsible for it. And again, it's going to be basically 2 lines. So these are, again, the heading is development of specific cell types. Of specific cell types. Here's the big topic is chromatin remodeling. Chromatin remodeling. And with chromatin remodeling, remember, chromatin is what chromosomes exist as when the cell is not dividing. So in interphase, the chromosomes have not, the DNA has not condensed yet. The chromatin is very diffuse. So when the genes have to be turned on and off, even in the absence of cell division, right? Because the cell either needs to prepare to replicate its DNA, or it needs to prepare to divide. So there's always gene expression going on. So this is the only thing that you have to remember, that there are 2 types of proteins that we'll discuss really, or mention. There are, and these are named in Drosophila where they were first described. So let me, excuse me. So this is what you need to remember. There are trithorax proteins, excuse me, and there's polycomb proteins. And these are named after Drosophila where they were first described. Trithorax lead to gene activation, and polycomb lead to the gene repression. So trithorax will allow the chromatin to become more accessible, so the genes now are active. The polycomb will tend to make the chromatin more condensed, so the genes are not accessible, they are repressed. Right? And here's another mnemonic that will help you. It's actually the same mnemonic, PP, right? Polycomb, repression. You can come up probably with a better one, but that's how I remember it. Questions about this part? And again, that's the only. There's a few other topics from this module before we go into other bigger topics. So there is also alternative splicing, so which I talked about. So please remember that. And alternative splicing is, let me use it in the context of a true-false. Alternative splicing, right? The idea of the concept of alternative, the process of alternative splicing, let's put it this way. The process of alternative splicing will generate different mRNAs from the same DNA. True or false? Different. Well, actually, let me phrase it differently, because you could argue this one both ways. But anyway, so alternative splicing is generating different polypeptides from the same mRNA. True or false? True, right? Because alternative splicing is basically doing what? You are leaving somewhat, you're putting the different exons together, right? That's alternative splicing. Because remember, splicing is the removal of introns and the splicing, putting together of exon. So in alternative splicing, what happens is that you get different mRNAs from the same sequence, because now you're going to get different proteins, Right? So this is, let me just quickly summarize it. Alternative splicing. So basically this is your mRNA, right? In mRNA, you have introns and exons. So let's make it simple. Let's say you have exon 1. Here's another, this is not directly on the exam, but true or false. The number of introns is greater than the number of exons in a particular gene. The number of introns is more than the number of exons. False. Are they equal? The exons are always going to be one more, right? Hopefully concept tells you that, right? Logic tells you that. Exon is expressed, right? So if you have exon 1, exon 2, exon 3, you're going to have only 2 introns. Intron 2, intron 3, right? Because you're always going to start with an exon, you're always going to be ending with an exon. Anything in the middle will be introns. So the number of introns is always 1 minus the number of exons. So let's make it actually a little bit bigger. So let's say you have 5 exons. So you're going to have 4 introns. In fact, in graduate school, way back when, I worked with a gene that exon 1 was only the start codon. So you had AUG, which is the start codon. That was exon 1. Then you have a huge intron, and then you had the other exons in quick succession with other introns intervening. But you're always going to get 1 extra exon, then you have introns. So what does alternative splicing mean? Alternative splicing means you can get different exons, and they will still be functional. That doesn't mean every single case. But you can get a protein that is E1, E2, E3, E4, and E5. So you put everything together. This would be one protein. You could get another protein that is only exon 1, exon 2, and exon 5. You could get another one that is 2, 3, and then 5. You could get one that's just 3 and 5. You could get one that's 1, 4, and 5, etc., etc. That is alternative splicing. And unfortunately, when it comes to human genes, there are some cases where this can lead to genetic diseases. So if the right exons are not combined, if there's a mutation that doesn't put the right exons together, that could lead to a genetic disorder. And if you were to take other courses in the sequence, you'll discuss that. Questions about alternative splicing? Okay. Stability of mRNA is the last small section in this grouping. So let me just talk a little bit about that. So stability of mRNA. Excuse me. And that is a small section. So here, there's only 2 concepts that you have to remember. One is the length of poly A tails. And 2, what are called destabilizing elements. Destabilizing elements. With the length of the poly A tail, it's simple. The longer the poly A tail, the more stable the mRNA is. Right? So stability here is directly proportional. Okay? The longer, the more stable. Right? Destabilizing elements, they're actually, just like the term suggests, these make it less stable. There's 2 features of them, please remember them. Typically, they find on the 3' end of messenger RNA. So they find on the tail end, if you will. And they're usually rich with AU. So the sequence is rich with adenines and uracils, and it's typically found on the 3' end. So when these 2 cases happen, the mRNA is less stable. Oh, we get richer. One is fine, frankly. I'm erasing because the kind.
Okay.
Yeah.
Thank you.
Yeah, thank you, though. Thank you. And I'll reciprocate by giving you a shirt.
No.
I would have used that one to erase anyway. Questions about this? Okay. So that's really the small parts here and there. Splicing. Okay, so another big section is trinucleotide repeat diseases. This was a middle section, if you will, and then there was a lot of elements there. So this is another big grouping, trinucleotide repeat expansion diseases, really. So they're called trinucleotide repeat expansions. or trinucleotide repeat diseases. So this is a big category. So here you do have to remember that we're dealing usually with 3 letters that are repeated over and over and over again, right? So 3 DNA letters or RNA letters represent a codon, right? So typically they can be found in the coding sequence or outside of the coding sequence. Right? What are you do have to know the examples of the trinucleotide repeat diseases that I talked about, right? We only discussed 3. There's about 30 of them. We only discussed 3. What are they? We talked about the most famous, which is Huntington disease, right? We talked about Fragile X syndrome, and there's one more. Start with M. Myotonic dystrophy, right? Myotonic dystrophy. There are type 1 and type 2. I have several tables in there, but for this grouping, please remember the 3 and then what are they? So for example, Huntington disease is found in the exon. Fragile X syndrome, the mutation, and by the way, the mutation is CAG, it's polyglutamine. For myotonic dystrophy, for Fragile X syndrome, it's found in the 5' end, so that's non-coding. And for dystrophia myotonica, it's found in the 3' end of the mRNA, which is also non-coding. Now, what is the hallmark of these diseases? There's a term that we described, which means they get worse generation after generation. It is called, anticipation, right? So you've answered 2 questions. Can you please also, if you want the shirt, I mean, if you don't want the shirt, you don't have to. But does that make sense? So you have to remember anticipation. Let me actually go in order. These are called dynamic mutations. Right? These are called dynamic mutations. You have to know this concept. Dynamic means they keep increasing, right? They're changing. More usually they increase. And because of this, they are associated with a phenomenon called anticipation. Anticipation means they get worse generation after generation. How can they get worse? Really in 2 ways. They get worse, meaning the severity is more, the symptoms that is more. What's the other one? Exactly, the person gets them earlier. Okay. With myotonic dystrophy, for example, if you look at, actually all of them, if you look at grandma, let's say, or grandpa, right, they might show some symptoms, but then, the child, their child, the father, if you will show symptoms earlier, and the grandchild might show it at birth, or very close, because again, now they've inherited hundreds of these repeats. I will not ask you whether the number of repeats is 50 or 100 or 1,000,000, but do know again the big ideas. Anticipation lists the diseases. There's only 3 of them that we discussed. Huntington disease. We talked about Fragile X syndrome, and we talked about myotonic dystrophy. Right? So again, this is found in an exon, the mutation that is. This one is found in the 5' end. This one is found in the 3' end. These 2 are non-coding. This one is coding, meaning the first one. Right, and then do know the type of mutation that we discussed, right? So for example, if you recall, we talked about this is a gain of function mutation, right? This is a loss of function mutation, and this is a toxic gain of function mutation. So it's listed in your note. Okay, questions about this? Okay, yes.
Are we going to have to differentiate type 1 being kind of
No, I'm not going to ask you about type 1 versus type 2. But you do have to know also that they show parental bias. Mm-hmm. Some are worse if they get them from dad, some are worse if you get them from mom. But I'm not going to drill to that level.
Okay, so do we need to know the parental bias?
No. Well, you have to know that there is one, but not the specific parental. Like, Huntington is worse if you get it from dad, for example. Oh, one more thing. I mean, you're going to get, I think it's going to be 1 point, but this should be an easy one. You do have to know the difference between a mule, right, and a hemi, and then a tiger and a ligon. So that is basically a recognition. So remember, if dad is a donkey versus mom is the horse, what do you get versus the other way around? So please remember that, or please know that, or please both know it and remember it. So the last point from this is maternal inheritance. Okay, so let me just walk you through a few ideas. My maternal inheritance is an example of cytoplasmic inheritance. So, maternal inheritance is example of cytoplasm. And by the way, genomic imprinting and trinucleotide repeat diseases are based on genes in the nucleus. So this is really an example of a modification of nuclear inheritance. Cytoplasmic inheritance is different because now we're not talking about DNA of the nucleus, we're talking about the DNA of what? The DNA of the chloroplast or the mitochondria. So that is why these are called cytoplasmic inheritance. And in general, it's not always the case, in general, it's inherited from mom's side, so maternal inheritance. Okay? So that is the key thing to remember. You do have to know the preparatory material. So for example, I do expect you know the idea of the endosymbiosis, right? The endosymbiotic hypothesis / theory, which posits that modern day organelles are really prokaryotic in origin. This happened when an early prokaryote was engulfed by a eukaryote and slowly but surely lost the ability to exist on its own. Now we know that from all sorts of evidence, right? The fact that they have their own DNA, the fact that their ribosomes are similar, the fact that they start with the same amino acid really, the fact about the size is roughly the same as modern day bacteria. The fact that if you look at their DNA actually, forget about the size of the DNA or their size, if you look at the DNA, it's very similar in sequence to that of modern day, or at least some modern day prokaryotes. So you have to know that part. How many membranes they're covered with tells you, right? Because one is from the original, one is from the original host. But you do have to know also specifically what the origin is, right? So for example, for a chloroplast, the origin was a blue-green algae, right? Which basically was a purple bacteria. Specifically, what about the mitochondria? It was an alpha proteobacteria. So that part, please know. Okay, so then we talked about some examples. So let's go over. So you take, and then we're dealing with inheritance of flower color in certain plants. I'm sorry, I'm not supposed to. Okay, so right. So let's say you're dealing with 4 o'clock plants. This is the example we gave, and this is the cross. You're taking white times green. True or false? You're gonna get green. If you take a white plant, leafed plant. Remember, the flower is a different color. It's not about the leaf. If you take a white leafed plant times a green leafed plant, you're gonna get all green. True or false? Right, exactly. This can't be a true false because it could be both true and false. You have to know more, right? In this case, you have to know who's the mother, right? Because this is a chloroplast inheritance. And please note, while chloroplast inheritance in some plants can be inherited from dad, in some cases it's biparental, but in most cases it is maternal. So, and then if it's a 4 o'clock plant, we know exactly it's maternal, so we can't tell. We have to know who the dad or the mom is, right? So then if I told you this is now the mother and this is the father, so to speak, right? They're all going to be green, true or false? False. That is now going to be false. Why? Because they're all going to come from mom. If mom is white, they're going to be white. If you do the reciprocal cross, you're going to get what? Green. Because now you're changing the sex. Right? What about this? Mom is variegated and dad is green. So we're still doing the same. You're going to get all variegated, true or false? True or false? Why false? Who said false? How would you make it true?
Because variegated is like green and white, so the offspring should get either or variegated.
Exactly. with variegated means, in fact, you've probably seen them, right? There are some that are ornamental. You see a leaf that has white and green sectors. That is variegated. So when you have a mom that is variegated and a dad that is, regardless what it is, you're not going to get variegated. You're going to get variegated and white and green at the same time. So some plants will have variegation. Some plants will be white. Some plants, or some leaves at least, will be green. And that has to do with the random segregation of the chloroplasts. Okay? So the answer here is false. It's going to be this plus green plus white. That would make it true. Right. You also have to know the concept of homoplasmy and heteroplasmy. Right? So let me give you an example. For example. Well, before we do that, you have to know the mitochondrially inherited diseases, right? So we only talked about two of them. I think one was M.E.E.T. There's probably about a couple of dozen. We only talked about two. The famous one is Leber hereditary optic neuropathy, LHON. Leber, which was the German physician who first described it. Leber's hereditary optic neuropathy. In fact, that's the other one was M.E.E.T, which is a long one. In fact, I probably would just test you on this one. This is a mitochondrially inherited disorder. Right? So with that in mind, first of all, any questions before? I mean, just to be on the safe side. The other one is called M.E.E.T. It stands for myoclonic epilepsy and ragged red fiber disease. The mitochondria really look misshapen. So myoclonic epilepsy and ragged red fiber disease. These are the two mitochondrial inherited diseases that we described. So, let me give you examples, and then we'll go from there. Problem examples, if you want. So here, the thing that you have to remember from, in fact, it's on the homework. So ideally, you remember what it stands for. When it comes to pedigree analyses, you have to remember that a square is a male, and then a female is a circle. And if they are affected, you're going to be shaded. And if they are not affected, they're going to be unshaded. Okay? So let me give you an example, and let's see how can it be explained? Mitochondria or not? That's the only thing you have to explain. They're all male. It's a big family. Now you can actually calculate the odds of this happening, right? In fact, let me ask you this. What are the odds of this happening? They have 9 boys in a row. What is the probability of a family having 9 boys in a row? Absence of twinning. There's no twins involved. It'll be, you don't have to come up with a number, just come up with a factor or a value. There's 9 of them.
Oh, there's 9.
Yeah, so it's basically 1 over 2 to the power 7. So again, you'll get a shirt if you want, because you've answered more than one question. Does that make sense? It's basically half times after the 7. But anyway, okay, so can this be mitochondrial inheritance? Why not? Mom gave it to all of her children. It doesn't matter if they're boys or girls, right? Now, if I throw a wrinkle in it, so now the last born, let's say, married, and they had a child, a girl that is affected. Can this be mitochondrial inheritance? It can still be mitochondrial inheritance if what is assumed? Not leakage, you're talking about leakage. No, that would be actually a good reason. But remember, we talked about homoplasmy and heteroplasmy, right? This child while extremely rare, it could be heteroplasmic, meaning they have some good mitochondria and some bad mitochondria. And just by chance, right, they, actually in this case, never mind, hold on. Let me, actually paternal leakage would explain it better. So the point I was going to describe, so let's carry this one to its logical conclusion, right? So the answer here, can this be possible? Yes, in the very rare case of paternal leakage. The point I was going to, and I conflated the two, let's say you have a different situation where it's the same family, it's a large family, but instead of this happening, they have 2 more kids, right? And they're both female, let's say. It doesn't really matter at this point. They could be both male or female, but they're not affected. Can this be mitochondrial inheritance? And the answer is yes, because mom could be heteroplasmic, meaning she has some good mitochondria and some bad mitochondria. So just by chance, these 2 kids inherited an egg, or were formed from an egg that had much more of the wild type normal mitochondria. So that is a phenomenon called heteroplasmy, where it's not the same mitochondria that explains them, okay? But if I told you that she's homoplasmic, meaning she has only one type of mitochondria, this is absolutely not mitochondrial inheritance, because she would be expected to give it to all of her kids, regardless of gender. Okay? So does that make sense? So again, assuming she's homoplasmic, not true. Assuming she's heteroplasmic, possible. Okay? Okay, so for the last, for the last, 30 plus minutes, I'm going to focus on population genetics. But first of all, any questions from the Module 8? Now, remember, Module 9 is population genetics. It's only one chapter. And again, I'm only going to focus on the notes. You are not going to be responsible for anything from the textbook, other obviously than the problems assigned, because they'll help you do better when it comes to problems on the exam. So when it comes to that part, I've worked out a few problems for you, and then these are really prototypical. You will not be expected to answer anything that is totally different from what we've described. Okay? And then similarly for population genetics, you're going to get roughly probably more actually problems from this part than concepts, because the concepts are relatively few in population genetics. So let's start with the concepts. What are concepts that you are expected to know? You have to know some definitions, right? What is a population? What is a gene pool? What does Hardy-Weinberg, what are the assumptions of the Hardy-Weinberg principle, right? Hardy-Weinberg law. So for example, true or false, Hardy-Weinberg applies to prokaryotic organisms in general. Right? It doesn't, because prokaryotes are not what? Well, no, because you could still work on separate species.
They don't binary fission, they're not sexually reproducing.
They're not sexually reproducing for the most part, and they're really usually not. You have to have a diploid species, right? You have to have 2 contributions. So you have to have sexual reproduction. So what are the other assumptions? The population needs to be large. Now, how large is large? I will tell you if it's large or not, but you have to know whether that large is an example of an assumption, right? It has to be sexually reproducing. It has to be no mutation. There's no migration, and their mating is random. That is key. Right? The mating, in fact, one of the problems on the homework is asking you about, whether you have inbreeding and outbreeding. So it's related to this concept. And before I forget, if you have, I'll stick around after if you guys have specific questions about the homework. So that is another part of the concepts that you'd have to remember. You also, and again, I'm just gonna put them all in one, and then we'll do the problems. You have to know the idea of also what is genetic drift, right? For genetic drift, and again, if you were to create flow charts or whatever concept maps, the idea of genetic drift, right? We talked about basically 2 big ideas, right? We have the founder effect, and then the bottleneck effect. And then the other one was basically a small size that stays small regardless. The difference between those two, right, is very important. With the founder effect, you're starting a population, you're founding a population. The founders, if you will, are not representative of an entire group. So that just by chance, you're going to have allelic frequencies change. Okay? The bottleneck effect is due to what? There's a natural disaster or human-made disaster or whatever it is. It's an infectious disease, it's an earthquake, it's a typhoon, it's a famine that kills a lot of the population, and what is left behind is not representative of the total, right? Meaning the original ones. So in both cases, you're going to get genetic drift. You're going to get variations in allelic frequencies that are not representative of the whole. So this could lead to really one of two things, right? You're going to get to a fixation, or you're going to get the loss of an allele, right? So genetic drift could ultimately lead to an allele being fixed, or to an allele being lost, right? So true or false, when an allele is fixed, it reaches 0, meaning 0%. False, right? It reaches 1, because now everyone has the allele. There's no other allele present. When it's lost, it gets to 0 frequency, right? 0%. True or false? When an allele is lost forever from a population. Forever is a big word, right? Well, it's a small word, but it's really a big meaning. It's false. Why? You can restore the allele in one of two basic ways. Either by, either by migration, someone brings the allele back into the population, or by chance, which is mutation. Exactly. And then again, you've answered more than once. If your t-shirt willing, please come. Does that make sense? So it's not lost forever, because you can bring it back by, you don't have your camera this morning. Oh, you do have it. Okay. So please, again, if you're kind enough. Right. So it's either by mutation or by migration. Now, again, another couple of concepts, and then I'll focus on problems. Concepts, when it comes to non-random mating, right? For Hardy-Weinberg to apply, you need random mating, meaning mating according to chance. Not, if it's not random and based on genotype, what is the phenomenon called? Well, if it's based on genotype, the phenomenon is inbreeding or outbreeding, right? Inbreeding, which is they're much more closer than what expected by chance. So basically, mating between relatives. If it's outbreeding, they are totally different from each other, family speaking. And if it's based on phenotype, what do you get? Positive assortative or negative assortative? And again, this is another and if you haven't tried it, it does make a big difference. Write things down because you remember them. So when it comes to non-random mating, right? Non-random mating. So it could be based on two things, genotype or phenotype. Genotype, you can have either outbreeding or inbreeding. Phenotype, you could have either positive, assortative mating. It's called assortative mating. It's either positive or negative assortative. Assortative. So out of these four, where does consanguineous mating, now this is a term that hopefully you remember from other, this is just on the side, I guess. Where does consanguineous matings fit? Inbreeding, right? Because consanguineous means mating between relatives. So technically it falls under the big umbrella of inbreeding. And again, inbreeding doesn't have to be, sick breeding, right? Where a father mates with his daughter, right? So inbreeding could be marriage between relatives, first cousins. And that would be an example of an inbreeding. You're not going to be responsible for calculating inbreeding coefficients, etc. Okay? Okay, so with that in mind, does anyone have a problem with concept? Because we'll go into some problems. And honestly, the problems fit into a few basic types, and I'm going to go over them. And the homework also gives you some more example types. So let me start with, you're dealing with a population of plants, and Hardy-Weinberg Equilibrium is in play. And the plants are segregating one gene with 3 alleles. So we have plants that we're dealing with A, which codes for flower color. And we have 3 alleles. And we have A1, A2, and A3. So, and the population of plants, well, actually, let me go over. So if this is the case, right? This is, again, not going to be on the exam, but, just to see how many of you remember. If this is the case, how many genotypes are possible? Assuming, again, Hardy-Weinberg, so you're dealing with a diploid species, right? How many genotypes are possible if this is the case?
8.
Not 8. It's not 2 cubed, right? Why 8, right? Use logic. What would be an example of those 8? Let's say there are 8. Give me an example of those 8. A1 would be an example, right? What's the next logical one? A1, A2, A1, A3, A22, A23, A33. There's only 6, right? So these are the ones. They could be either A1, right? They could be either A2, A2, A3, A3, or A1, A2, A1, A3, and what's the last one? A2, A3, right? So I'm just putting them in order because I have a question to follow. So it's A1, A1, 1, 2, 1, 3, 2, 2, 2, 3, 3, 3. So let's say you have, again, arbitrarily, 30 of these plants, you have 20 of these plants, you have 50 of these plants, you also have 50 of these plants, you have only, let's see, you have only 10 of these plants, and then 40 of these plants. So 30, 20, so 30 A1, A1, 20 A2, A2, 50 A3, A3, 50 A1, A2, 10 A1, A3, and then 40. Now, if my math is correct, these add up to 200. Yes, 200, yes. Okay, so here's the question. Now, we did not go over this directly in class or in the cloud or whatever, but you are expected to be able to answer this question. So here's the first question. What is the frequency of the A1 allele in this population? What is the frequency of the A1 allele in this population? And remember, with allele frequencies, if it's only 2, we use P and Q. If there's a 3, we add an R, and so on and so forth, okay? So here we have PQR, if you will. What is the frequency of A1? What is the frequency of A1? And then here, hopefully you guys will remember this, right? Forget about population genetics. Forget about population genetics. If I were to ask you this question, which I am, what is the frequency of seniors and seniors by status in this group, right? What would you need to know? Or in fact, you would need to know the number of seniors, and you would need to know what? The total. The frequency of anything is anything divided by the total of that thing. Okay? So what is the frequency of A1? It's simply the number of A1 alleles, right? Divided by the total number of alleles. The total. Does anyone have a problem with this? Now, what is the total number of alleles? 200, true or false? It is false. It's not 200. It is 400, right? Because each plant has 2 alleles. So if you have 200 plants, you have 400 total. So the denominator is very easy. It's 2 times 200. Now, what is the numerator?
It's 2 times 30 plus 50 plus 10.
Right. So basically, the number of A1 alleles is how many A1 alleles you see. A plant, that is A1 has 2 alleles. So I'm going to take 50 and multiply it by 2. A person that has a plant person that has 2, right, has 0. So I'm taking this by 0, this by 0, this by 1 because there's only 1, this by 1 because there's only 1. So the answer is going to be 30 times 2 plus 50 times 1 plus 10 times 1, and you just do the math. I mean, it's almost like calculating your GPA, right? If you take a 3-credit course and you get an A, now you obviously have 4 times 3. But if it's only a 2-credit course, then obviously your factor is not 3 anymore, it's 2. But the 4 is still there. And then you just average them out. And then you do the same thing for A2, you do the same thing for A3. Now let's change the question. What is the frequency of A1 plants in this population? What is the frequency of A1 plants in this population? Now the answer is what? Now the answer is 30 divided by what?
200.
Now 200. It's not 400 anymore because you're not counting alleles, you're counting plants. So the frequency is now simply 30 over 200. Questions about this type? Okay, now there is, let's do Hardy-Weinberg Equilibrium. This is counting alleles. And now remember, with counting alleles, I went over 2 methods. One is counting alleles, which is this one. The other one is using frequencies. So if you're given the numbers of individuals, you use this method. If you're actually only given frequencies, then you would use the one with the frequency. Okay, so we did both. So let's do this example. Right, I'll start with a basic and then we'll go over more increasingly difficult ones. So let's start with the simple one. We have one gene, A, that's segregating into alleles. So gene A has either A or little a, and you're told that the frequency of big A is 0.2. What is the frequency of heterozygotes in this population? What is the frequency of heterozygotes? Now remember, heterozygotes are A with little I, right? So this is one that, please remember, at its heart, the Hardy-Weinberg is very easy. Okay? It's P squared plus 2PQ plus Q squared. P squared, while not law, by convention, we use it to represent the big A. Q squared, we represent the 2 little a's, and 2PQ is the heterozygotes, which is A little a. So really, P plus Q all squared is this one. So the Hardy-Weinberg is P plus Q all squared, which gives you P2 plus 2PQ plus Q2. Right? So if A is 0.2, that means little a is 0.8 because they have to add up to 1, which means 2 times 0.8 times 0.2. So that'll be 1, whatever that answer is. So this implies the frequency of little a is 1 minus 0.8, which is 0.2. So the frequency of A little a is 2PQ. It's 2 times 0.2 times 0.8, which is 0.32. Right? So 32% of this population is expected to be heterozygous. Right? So this is 2PQ. 2 times 0.2 times 0.8. So the answer is 0.32. Questions about this? Okay, let's do another one. Again, just to show you that really they're all the same idea. As long as you get one, you should be able to get to the other. Let's do one with diseases, right? I'll tell you, for example, albinism is an autosomal recessive disorder. And in a particular population, so we're dealing with albinism, you're told that it's autosomal recessive, and a particular population, you're told that 1 in 10,000 people, let's use this, 1 in 10,000 people has albinism. What is the frequency of the normal allele? What is the frequency of the normal allele? Right? So the fact that it's 1 over 10,000 is, well, let me rephrase. 1 over 10,000 is equal to what? Is equal to Q squared, absolutely. Because remember, the recessive phenotype is the little a, they would have to be albino, right? To have albinism. And that is represented by Q squared. So Q squared is 1 over 10,000. That's the answer right there. Q squared is 1 over 10,000, which implies that Q is what? Root 1 over 10,000, which implies that Q is what? 1 over 100. 1 over 100. Because remember, if the square root of 10 to the 4 is 10 squared, the square root of 10 to the 8 is 10 to the 4. So Q is 1 over 100. What is P? 99 over 100. Because it's 1 minus Q. Right? What is 2PQ? 2 times this times that. Does anyone have a problem with that? That's another side of a question. Let's do one with X-linked inheritance. Let's do one with X-linked inheritance. Okay, so same thing. We're dealing now with, let's say, hemophilia. So I'll tell you that hemophilia, so now we're dealing with hemophilia. Hemophilia, you're told that it's X-linked recessive. While hemophilia is not compatible with long-term survival and reproduction, especially some forms of hemophilia. So yeah, let's say it's hemophilia. Actually, that's not do hemophilia. That's not very correct. Let's do colorblindness. So colorblind, same idea. Colorblindness is an X-linked recessive trait in humans, and I'll tell you, for example, in a particular population. 3% of males are colorblind. So in a particular population, 3% of males are colorblind. What percentage of females are colorblind? If you're told that the percent in males, can you calculate the percent in females? Because remember, it's X-linked recessive. So that means for a male to be colorblind, what would he have to have? Genotypically speaking, just one. So basically, a male to be colorblind, he would need to be X little c with a Y, assuming C is the colorblind allele. For a female to be colorblind, what would she have to be? X little, X little. So basically, the 3% is equal to what? What letter or letter combinations would give you 3%? If you're using the Hardy-Weinberg notation. Sorry?
Q.
I'm sorry? Someone said it. Q, right? It's going to be Q. Well, someone said Q, so take credit for it, whoever you are. Or even if you're not, take credit for it. And Q. Q is 3%. So what is the answer in females? Using letters as well. It's going to be Q squared. Right? So it's going to be 0.03 times 0.03, which is going to be rarer in females, right? So Q is 3%, which means 0.03. So to get to the female, it's Q squared, which is 0.03 all squared, which is 0.0009. That's what explains why it's very rare in female. Okay. So any questions about this? Okay, I have two upper level sort of problems, and I can't stress it enough. There are in each chapter, in each module, I indicated what are called concept checks. These concept checks are frankly from old exams, so be able to also solve these because they include examples of the nature that I'm describing. So let me go over one more, and then we'll finish. Actually, two more that are increasing in difficulty. Okay, so here's the first question. Let's make it easy. Can anyone come up with any fictitious trait? Something you always wanted to? Anything, any weird thing that you can think of, any trait. Sorry? Eyes, okay. So we're thinking about eyes. Can someone give me two different shapes? Sorry?
Brown and blue?
Oh, color. I was looking for more esoteric. Okay. No, that's actually, I want the fictitious one.
Purple.
Shape. Let's do shape.
Oh,
So square, thank you. Okay, square eyes, and the other one would be wild type. Okay, so normal, which is round or oval, whatever. So assuming that this is a trait and the gene is called E, right? And big E is wild type and little e is square. Assuming it's a single gene inheritance. And you're dealing with a population that is under Hardy-Weinberg Equilibrium. Okay? And then you're told that 64% of the population has normal eyes. What is the frequency of big E little a? So again, you're told that 64% has normal eyes, which is the wild type, which is the dominant one. What percentage is going to be expected to be heterozygote? Now, in all of these, right, in all of these, and I think I mentioned it in the lecture, in all of these, you're going to be given enough information to get one of them, and then the rest will follow. Now, if you have two of them, I have to give you one of them, right, to calculate the other one. If you have three of them, then I have to give you two to be able to calculate the third, or give you enough information. So let's do with that, work with that one. Can anyone suggest an easy way to do it?
Find out what it is in little e, so you can use that as Q.
Right, but how can you do that?
0.64, like 1 minus 0.64.
Exactly. Here's the problem. You can't start with normal eyes, right? Because normal eyes has this. Can either be big, big, or can either be big, small. That is two unknowns, right? I can't figure it out. I can't have two unknowns and determine them simultaneously, right? If I tell you two numbers add up to 10, they could be literally an endless number. It could be 0.1 plus 9.9, it could be 5 plus 5, it could be anything, right? But if I told you you have two numbers, they add up to 10 and one of them is 5, or one of them is 6, the other one's gonna be 4. So that's exactly what I'm doing here. I can't start out with this one, it's too unknown. I'm gonna start with the exact opposite. 100 minus 64% is equal to 36%. That 36% is equal to what? Q squared. So what is Q? From B to B.6. Now here's another thing that I'll tell you from now, which will help you. On these problems, I'm not gonna give you a number where if you want to calculate the root, you have to take your calculator and put it sideways because it doesn't end, right? You're gonna get a number that is pretty round, okay? So you might get 0.6, at best you might get 0.652, for example. But if your answer is 6.652, 69324, whatever, it's almost like pi, it goes forever, then you're way off. You're gonna get a nice round number. That is why the numbers I'm gonna give you are either 64%, or 91%, because the other one would be an exact quick root. So that said, Q squared is 0.36. So therefore Q is root 0.36, therefore Q is 0.6. If Q is 0.6, P is 0.4, EE is 2 times 0.4 times 0.6. Does anyone have a problem with this? Any problem with that? Okay, so let me finish with the hardest question. And so for this, we're going to deal with 3 alleles, same thing, and we're dealing with blood grouping. So we're dealing with, and which is very similar, at least, the idea is to the homework problem that you were given. Okay, so we have a population, and you're told that the frequency of IA, actually IB. The frequency of allele IB is 0. Let's say 2. And the frequency of blood type O is 25%. I'm just mixing it up. This is 0.2 plain. This is the frequency of blood type O is 25%. And the question is, what is the frequency of blood type, blood type A? So let me rephrase. Here's the question. You're told that you have a Hardy-Weinberg equilibrium, or at least we're dealing with a population that's large enough, and you're told that the frequency of allele IV is 0.2. You're told that the frequency of blood type O is 25%, and you're told to determine, or you're asked to determine, what is the frequency of blood type A in this population, right? And while an answer could be, we need more information, that is not the case. You have all the information you need, okay? So for these type of problems, here's again a big thing to keep in mind, and then the rest will follow. You cannot solve equations, let me rephrase. You cannot solve unknowns unless the number of unknowns is equal to the number of equations, right? You probably know that from other courses. I can solve an equation in one unknown. I can solve 2 equations in 2 unknowns, 3 in 3, 4 in 4. I cannot solve an equation if there's more unknowns than there are number of equations, right? So going back to, x plus y is 10. You have an endless amount of solutions. X plus 5 is 20, you can solve x. So here's the same reasoning. I have 3 unknowns here, right? I have IA, I have IB, and I have IO. I have enough information to get 2 of them. I'll get the 3, and then I'll work it out, okay? So one of them is straight off the bat, right? I don't need to do anything. It's already there. So check. Okay. I was told that the blood type O is 25%. Is that enough for me to get IO? Of course. Because you can only be blood type O if what? If you are IO-IO. So therefore, IO-IO is equal to 25%, right? So the frequency of IO-IO is equal to what? 0.25. So what is the frequency of IO? Root 0.25, right? You're gonna square root both sides. So therefore the frequency of IO is what? 0.5. That's a weird find. Does anyone have a problem with this? I know IO, I know IB, I can calculate IA. So what is the frequency of IA? It is 1 minus the others, 0.2 plus 0.5, so the frequency of IA is 0.3. Does anyone have a problem with this? Frequency of IA is 0.3. Now, this is the point that I was trying to make earlier. If you're doing problems like this, and instead of getting 0.5, you get something that keeps running, right? 20 digits, you're way off. You're doing something wrong. So I'm not there yet, but I'm almost there. So what is the frequency of blood type A, right? I'm now looking at blood type A. So to be blood type A, what does the person have to be? This is, again, material that you have to know from previous right? To be blood type A, what do you have to be? I1, IA, right? And IA, IO. Sorry. So what is IA? It's basically 0.3, what? Times 0.3, right? Because you want this and that. What about IA-IO? IA is 0.3. IO is 0.5. Am I done? Times 2, exactly. Don't forget the 2. The heterozygote always comes with 2. And it's actually very easy. It's basically based on a Punnett square, right? So for you to be a homozygote, big A, there's only one way. Dad has to give you a big A, mom has to give you a big A. But if you're a heterozygote, there's 2 ways. Mom can give you the big A, dad the little,
or mom the little, dad the big. That's why there are two ways to get to a heterozygote. So don't forget, you have to multiply it by 2, regardless of the heterozygote. And then I multiply, true or false? Why do you add?
Because it's the or.
Exactly, it's the or rule. It's not this and this, it's this or this. So the final answer is actually the addition of the 2. So it's going to be IA, which is 0.3 squared plus 2 times 0.3 times 0.5. You add them and that will be your frequency. Right? So I'm too old to beg, but I'm going to finish with 2, please. Please, if you are here, donate your notes, if you're kind enough to donate your notes. And then 2, if you haven't done the assessment, the student assessment of instructions, please consider doing that, because it does make a difference. It goes into our file, good or bad. And I'm going to stick around if you guys have any questions about the homework or otherwise. And again, if you were claiming a t-shirt, please come over. And then please claim a banana.